Good afternoon, everyone, and welcome to the November Food for Thought seminar, which is our final one for 2025. I'd like to start by acknowledging the traditional owners of the land on which we meet today, the Whadjuk people of the Noongar Nation, and recognise their ongoing connection to this land and their contribution to the life of our city and state. I pay my respects to elders past, present and emerging, and I extend that respect to any Aboriginal and Torres Strait Islander peoples joining us today. Please take a moment now to turn off your mobile phones or put them to silent. Before introducing today's speaker, I'd like to share a few quick updates from the Foundation. Our new coffee shop Panacea, operated by Levine, is now open. Feedback since opening has been excellent. If you haven't tried our coffee yet, I highly recommend it. The courtyard and lobby are so close to completion. Unfortunately, we couldn't get the two to quite coincide, but I'm hoping that by next week you will be able to enjoy the courtyard while you drink your coffee. And so everything is coming to fruition very quickly now. I do know that it has been difficult with entry to the building being via the car park, and I really want to thank everyone for their patience during construction as we work to bring you this revamped site. And just to let you know that we'll probably plan a formal launch event sometime in the new year. I would particularly like to acknowledge my staff member Troy Morse, Head of Facilities, and also Zoran, who works with him, for their tireless efforts in bringing this project to fruition. It's been enormously stressful for them. It's a huge workload they've taken on, and there's going to be no let up because we're following straight away with the installation of the lifts, and following the lifts, the new PC2 laboratories. So please be kind to them as they work to bring you all these fantastic changes. We're also delighted to announce that next year we are going to launch a new Career Advancement Fellowship program. Details will be provided via our website, so please do look out for that. Finally, please also look out for new details on our Food for Thought series for next year. And I want to thank all of you who have attended one or more sessions throughout 2025. It's now my great pleasure to introduce today's speaker, Professor Alexei Tursky, a Future Health Research and Innovation Distinguished Fellow, Head of the Chromatin and Aging Laboratory at the Harry Perkins Institute, and a Professor at UWA. He also holds a part-time research position in San Diego. He completed his PhD in molecular immunology at the University of Lausanne and postdoctoral work at Stanford. Professor Tursky established his own research group in Switzerland before relocating to the Burnham Institute in the US, where he investigated epigenetic mechanisms of neurogenesis, which led to new insights into Zika virus treatment and the mechanism of viral propagation. His team pioneered a new imaging technique, MEAL, which maps epigenetic patterns in the cell nucleus. This evolved into IMAGE, a groundbreaking biomarker that measures biological aging at a single cell level. Many thanks once again to Professor Markus Schleich, who will facilitate the Q&A session and officially close out our event today. Please now join me in welcoming Professor Tursky and his presentation, Imaging of Chromatin and Epigenetics Reveals Cell Identity and Biological Age. Thank you.
Thank you very much for inviting me. Truly appreciate. I didn't realize how intimate the room is, so I had a pointer, but I probably don't need it. I will point with my finger. Let's see. I'm not sure if that's something we can remove it. All right. So, some people may have heard that. Okay, it works. Wait, no. So some people may have seen this slide before. I gave a very brief talk at FH Fiery Day, last week I think was it. So there'll be a few slides if you see it. I realize only 2 switches here on and off, so it's probably as dark as we possibly can get the screen without plunging everybody in complete darkness. So what helps people live long? I probably, yeah, I know people. I don't want people to miss the food. Live long and well, and an important question, right? Which we all deeply concerned with. So how do we know? Well, one thing that I think is really critical is the answer to this question is very likely to be different to everybody in this room, let alone outside of this room, right? And that's just a quick example. The slide I stole from Michael Schneider when he gave a talk at our place, which really impressed me. So it's a normal individual temperatures. You can choose Fahrenheit or Celsius, up to you, but you realize that people at the bottom and the top sustain their normal life at a condition that otherwise probably will require immediate emergency room, right? Immediate hospitalization. But these people living at 33 and they're 38 and they don't need hospitalization, at their normal baseline temperature. If you think about it, it's quite staggering. So this is not even a response to treatment, they're just a baseline. And I think a lot of researchers, doing mice research in particular, when I see this mouse, it's just, everything is great, but that mouse is just up there, and then it's an outlier. Well, you cannot call it an outlier. And so I think that variance is not a bug, it's a feature of the system. And to me, actually, the most interesting feature, and you'll see in a talk why I think this is the most interesting feature. And essentially, that's what we should study. Now, what make people and how can help them live long and well? Well, there's a lot of challenges. I outline here several of them, which I think are more or less familiar to everybody. And one that I'll talk a little bit more about, of course, is that we still measure aging as a chronological age, which is a terrible way to operate because there's a lot of variability, as you probably already know, in that measurements. Now, the path forward really would include a number of changes, in particular personalized regimen to longevity, because everybody convinced now the medicine should be personalized. Well, longevity intervention should be also personalized. Not all people are going to benefit from dietary restriction or ketogenic diet, even exercise. And so what I'm particularly concerned is that measurements of aging should really be done on single cell level and include the measurement of biological age, which is kind of a fancy name for a functional age. You essentially need to tie whatever you measure to a function. And that function better be measured at single cell level. So I mentioned here some of the words that you probably heard before from Luigi Fontana in Sydney and many others. The idea of this chronic disease to chronic health and mechanism-based medicine is definitely not new. So what we try to bring from my lab to the field is really that little piece, and hopefully a lot of other things will fall in place. So alternative title for my talk is usually this one, An Image is Worth 1,000 Genes. And you'll see in a minute why I think that's probably true. If we look at human faces, most of us would fairly easily guess, a gender and age, race, and probably function. That individual probably has not as much function as this individual. How do we know? Well, our neurocomputer has been trained. All this is AI generated, so don't worry about those faces. Now, what if you can look at the epigenetic face of a cell, we call it, and tell something about cell identity, which is a cell type, cell state, and potentially cell function. That's a typical image that we get a combination of different couple of epigenetic markers, which is in this case will be chromatin, which will be histone modifications, methylation, acetylation. We're not going to talk about DNA methylation. So a lot of people, when we say epigenetic, mean translated as DNA methylation. DNA methylation is one tiny sliver, a tiny thing in the epigenetic landscape. So conventionally, how people were thinking about cell identity until now? Well, of course, from the time of Ramonica Hall, these beautiful images of neurons and Purkinje cell will tell you that one of the ways to look at and think about cell identity is using morphology. However, a lot of cells are kind of similar in shapes and form, yet are very diverse in function. And so these days, most common way to look at a single cell will be single cell RNA sequencing. All familiar with that, essentially propagated by the hugely successful Illumina method and instrument. However, there are a few limitations to this. And well, besides the fact that it's expensive and time-consuming, the fundamental is that it has no accuracy at single cell level. The people who know this are probably familiar that in order to call these cells, whatever it is, a B cell, this cell, you need to do pseudobulking. So you need 1,000 cells, actually, to look at any mechanistic functions, a mechanistic judgment whether the pathway will be activated or not, whether genes express or not. At a single cell level, not much you can say because of the dropout effect. If you didn't sequence that gene, it doesn't mean anything. It means that it wasn't there, but it wasn't there because you didn't pick it up, or wasn't there because it wasn't expressed. You cannot tell. So some time ago, this gentleman, Conrad Waddington, and probably people heard about the Waddington landscape more than Conrad himself, established this, essentially introduced the notion of epigenetic, the space embedded in a genotype and phenotype, which suggests that somewhere between the genome, this is a wonderful blueprint, but that doesn't really tell us what's happening at single cell level, and the proteome, which will be a fantastic way to assess the cell identity. There's a couple of layers, in particular this epigenome. The beauty of epigenome compared to these two is, a particular proteome, is that it encodes the present, but potentially future states. Probably people familiar with the bivalent chromatin domain that could be resolved in activation or repression. And so as much as you can tell whether genes express or not, you also can tell a future of that pathway or gene, whether it will be expressed. And so I would posit that this layer had more information eventually than the proteome, if you can read it accurately at the single cell level. Now, one thing that one thing that I like to stress particularly is that aging, which became very dear to our interest in my lab, a lot of time focusing on aging mostly exclusively, is that aging is a real epigenetic phenomenon. So we starting and ending with pretty much the same genetic material, given or taken somatic mutation, and some people argue they're very important, possibly, but they're random. So they're hotspots, but they're still random. So it's essentially the way you handle your genes. That genes expression and regulation is handled very well in the beginning and not so well at the end. So it tells you, if theoretically, if you're a Maxwell devil and you can have the wand and you can rewrite the epigenome just the way it was back 10, 30, 40, 50, 60, 70 years ago, then you'll get a young organism. Now, there's a lot of indication. Some of the high-profile paper, some people disagree with this particular paper, but the idea is that the epigenetic information, epigenetic probably really an important component, if not the causal in mammalian aging, and that's idea again is fairly old. This is not that much new, but some experimental evidence I think is coming up. I'll show you in a couple of slides that in addition to cell type, cell state, such an aging, we can infer actually cell function from epigenetic landscape. And that will be, for example, the motor function or the hearing function in inherent cells. And so that is to say that the ensemble of this chromatin epigenetic modification, so if you can see through in every cell all the epigenome and you can extract this data, which I'll show you how we do it, you probably can arrive at a functional state of the chromatin. The asterisk here to remind me that the functional state is extremely labile. It changes all the time. So what I'm going to talk about today mainly are snapshots. You take a picture. Although we are taking movies, and there's a way to do it in vivo in the live cells. And so that should give us all this phenotypes or function of the cell at a single cell level, and therefore we can achieve resolution that's needed, I think, for contemporary analysis of human health. So how we do this? Very briefly, so we're starting with an image, confocal image of the nuclei that labeled with different epigenetic marks. The high resolution results in a really good analysis. The better resolution, the better analysis. And then we translate basically that combination of voxels into the table. And the way to do it is using that cotensors, the coordination of chromatin epigenetic determinants platform that we developed. I'll talk more a little bit about it. We use some machine learning, and that will give us two things. The first is essentially a distance matrix. So you put every cell in a multidimensional space, very similar as you do it with the gene expression, right? You have 20,000 genes at 20,000 dimension. Here we have about a couple of thousand features. It's a couple of thousand feature dimensional space. And so you can measure the distance matrix, and therefore you can measure position and mutual relationship. And once you learn which cells are which, you can tell where your experimental and control cells are going. We also can learn cell heterogeneity, something I think is extremely interesting because that's provided some ab initio information about the system. You don't need to know the label to measure heterogeneity, although I'll show you in a minute. This is quite a complex story. I'll have a few slides at the end. So just to drive down what exactly we're doing, because even after a few years, my collaborators still coming and asking me, well, so what is the level of this mark? I'm telling you, it's not a level, it's a pattern. So what we're working with is patterns. So the epigenetic marks exist in every single cell. If you don't have a 27 trimethylation in the cell, that's not a cell, not a mammalian cell at least, or not eukaryotic cell, I should say, because these marks are conserved from Hydra to humans. So a few million years there. Now, what we look at a pattern. Now, I'll show you this too, and I call it markers versus patterns. Sometimes I'm jokingly show researchers versus pathologists. But basically, if you look at this slide, these colors, there's not much you can tell. It's just the colorful things. You probably think this is DAPI. However, if you look at this slide, if anybody from pathology department working with skin, well, you'll figure out this is skin, of course. This is cornified epithelium, this is stroma, this is somniferia, something growing in the stroma. And sure enough, you can reveal the label, but only revealing the label, you can tell anything about this slide, whereas H&E stain have an interpretation without any label because people get trained. So if you get trained on recognizing this pattern, you can recognize any fraction of this pattern. It's still the same. So that's what we're doing. We're recognizing the pattern, but in addition to one color, we have many colors, and patterns are three-dimensionals. All right, so how does help us to look at aging, in particular biological and functional age? You're probably familiar with this lady, Jeanne Calment, assuming to be the longest lived lady on the planet. It's hard to find her not drinking and smoking, usually both. Both contraindication for longevity. But remember, it's individuals, it's not average that we're looking about, we're interested in. Well, there's a lot of people that are following this, and there's some genetics in that for sure. This is my favorite, Brenda Milner at Still, faculty on McGill. It's her TED Talk at 93, lecturing 97, 103, delivering an award talk. So if you look at, so those are anything but averages, of course, like genetic lottery, you'd say, sure. Not all of us are going to win genetic lottery. But if you look at the average gain, that's about 10 years in the past century of the average lifespan. And that's, I think, what I'll be very much interested in talking about. It's not this we're interested in, it's this we're interested in. So comparing averages, what's on average happening, will give you this. Understanding individual features will hopefully give you that. All right. So again, looking at aging from the functional perspective, been occupied people's mind for a long time. And geroscience actually is real science. It's not a fake science. There have been meetings and whatnot for at least a decade. And the posits are very simple. So if you can understand mechanisms of normative aging, which is not an easy thing because normative aging and diseases are very, very closely related. But imagine you have a mechanism or a process that's relatively free of disease. Then what one can argue, if you slow or turning back the clock, if you affect those, impact those mechanisms. So there are two things happening. You increase resilience of the organism, because we all know the younger organisms are resilient. But you also reduce the risk of age-related diseases because of major conditions. Neurodegeneration, cancer, vascular are all age-related. Major risk factor is aging, nothing else. And so really, it makes sense. that instead of chasing every single disease in isolation, especially terminal condition, which is futile effort, a lot of attention should be paid on how to reverse the fundamental mechanisms of aging. And a very, very little gain in that direction will give you huge advantages in many diseases. Now, very recently, Vadim Gladish also turned the table, suggesting that, at least in this paper, that it's the oldest tissue, biological oldest tissue that fails, that became disease or malignant. So again, another interesting indication. So if you know how to measure it, you can probably predict which tissue will fail first. So you've probably seen this at many times. So we're talking how to measure it, right? We're all now convinced that we should measure somehow biological age. So how you do it? Well, there's famous biomarkers, molecular biomarkers of aging, and there are a number of those. And essentially what's happening is that you can measure anything you want on that list, and the list is long, and plot it against chronological age, and you can dissect it on the average. And then the distance to the average will tell you essentially the acceleration, the deceleration of biological age. Particularly, people do it a lot with DNA methylation clocks. So this person will be decelerated age, this accelerated aging. And it's all good and well, except, again, quite a time ago, those two gentlemen had a very good definition of aging biomarkers. And it's the one that in absence of disease, which is important and difficult in practice, the biomarker should better predict functional capability at some late age, better than chronological age. That's what you want, right? You can, there's no chronological age, but it's not good biomarker, not good enough yet, the best biomarker at the present for most of disease indications. And so what we argue is that out of all the relationship that you want to know, you can plot age versus biomarker or age versus function. What you really want to know, it's a biomarker versus function. And I think this is why it's important to do single cell resolution, because function is defined at the cell level. The fundamental unit of biological function is a cell. The cells then aggregate in a cell type, they form tissue and organs and interact, but the fundamental unit of function is a cell. You only can understand the function of an organ and tissue if you know what cells are doing, right? There's a B cells and T cells. You cannot blend B cell, T cell, and monocytes and ask what's the function of that cell. The function is, that's nonsense, right? That's a nonsense cell, no function. So you're familiar probably all with the DNA methylation clock, and essentially if you look at a CpGs and change of the CpG with age, one can select between 2 and 0.001% fraction of CpG that do change with age. And over the past decades, starting from Steve Horvath's, there's a lot of efforts in using the DNA methylation clocks as a biological biomarker of aging. And there's several efforts going in Singapore and in Boston and all over the globe. Now, it's all well and good, except the few limitations on fundamental limitations that up to today, this day, there's no single cell level analysis of DNA methylation clock. The few papers, they try to impute single cell from the bulk, but really that's not what's happening. Also, you can imagine that's only a one out of many epigenetic marks. So we propose essentially employ many different epigenetic marks, and using confocal microscopy, which is much easier and faster, to look at epigenetic age and single cell level. And there's a number of perks in particular that you can do it in high throughput and analyze cells in situ, which I'll show you in a minute what we do. All right, so a number of years ago now with Peter Adams, we essentially label PBMC from a mouse at different age. This epigenetic mark in DAPI, just, I apologize for immunologists, distinguish between CD3 negative and positive cells. And then when all these measurements that I just showed you a few slides ago were reduced to two-dimensional with the multidimensional scaling, very similar to PCA. If you're familiar with the principal component analysis, that's pretty much it. And then you plot every cross or dot here is individual mouse, or PBMC from that mouse. What you appreciate, well, there is separation between crosses and circles. This is T cells and non-T cell, but what's really apparent in the strongest kind of data set is that you see the trajectory. It basically goes from green to brown just in front of your eyes, right? And so then we thought, okay, well, if you travel along this line, which we essentially call the image, and plot it against time, well, you figure out that there's a correlation. Now, importantly, in this case, there's no regression, no elastic network on deep learning or linear regression, and that's because this extracts about 80% of variance from data set. So you don't need any regression. DNA methylation clock extracts about 2% variance of data set. So if you plot all DNA methylation, you'll just have a haze. There's nothing there. Now, if you extract 80% of variance, you don't need regression. You just plot it. And so the trajectory of aging basically emerges in front of your eyes. And that's true for many different organ tissue that we tested. A lot of this needs to be done at a single cell level because all this basically grinding the entire organ, it's terrible things to do, but we've done it. Have to apologize for that. Nevertheless, on the whole organ level, you can see there's an increased trajectory of this. Aging, of course, different marks can give you slightly different results, and it's all very interesting and probably completely meaningless because this has to be done on the single cell level. We looked at an intervention, and just to have a checkbox, the intervention known to slow down biological age indeed choose that doxorubicin make cells appear older, statistically significant canine remethylation, but the trend is in all marks. And with Ross Anderson, we did caloric restriction. Only caloric restriction is supposed to make organisms appear younger, and indeed the case here. And again, statistical significance on K39 and trends everywhere, but that's been done like on 4 mice. So it's pretty good for that amount of animal. We also look at a proportion of change basically goes along the aging axis versus orthogonal, and that's coefficient of determination. If you do this, then you appreciate that about half of the changes in both doxorubicin and caloric restriction interventions basically collinear with aging, and half are not. So essentially, there's a lot of things happening, and we'll know, but half of the changes that we can detect are indeed, at least with respect to our measurements, are age-related. So that's good. So that intervention, the biomarker is supposed to read interventions that reduce or increase biological age, and it does. So together with Manuel Serrano, we'll look at this very interesting technique of partial reprogramming. It's essentially expressing Yamanaka factors in vivo, but you don't go all the way, because if you go all the way, you want to try to arrive to iPSC, and you arrive at teratomas somewhere earlier. So, but partial reprogramming includes a cycle regimen where doxycycline injected like once or a couple of times, and so you induce OSKM, but then you stop. So essentially, always try to stay within the lineage. You don't leave the cell lineage. And that's examples from Manuel suggesting that there's indeed epigenetic change, methylation change expected in OSKM expressing mice. And so we looked with image at liver and skeletal muscle, and indeed you appreciate that all mice with OSKM appear younger on the image scale. If you look at the individual mice, though, you appreciate it's also seen here, but it's kind of dramatically exaggerated here. The heterogeneity is pretty staggering. So this is the same mice. Actually, all of these mice are the same chronological age. They're all old. And these mice been rejuvenated, and this mouse is supposed to be different from the same as this mouse, but they're different. This mouse seems to be exactly the same as the old, and that somehow seems to be rejuvenated, right? They're genetically identical. They all have the cassette, yet they're different. So what the different mean? Is this a fluke of the imaging, basically a technical limitation? Well, to answer this question, we collaborated with Amanda and asked a simple question. Can we connect that measurement with function? Because remember, any measurement, at least biological age biomarkers, makes only sense if you can connect to the function of something that you measure. Otherwise, it just becomes a number. So we use chronological identical mice exactly as in the previous slide. They're all old, about 23, 24 months old. We ran a number of behavioral and metabolic tests, the Rodorod, the metabolic chambers, and a bunch of others. We used the young and old as an age control, the reference point. Skeletal muscle being used because it's an easier thing to kind of, most of the locomotion behavior, of course, is implemented through the skeletal muscle. Molecular markers, and then we use a couple of things such as information distance and hyperbolic embedding. I'll particularly talk about hyperbolic embedding because I think it's a wonderful measurements, I should say, embedding or mathematical apparatus. Believe it or not, we live in a hyperbolic world. You're probably familiar with the Poincaré ball. So it's a space different from Euclidean, which is isotrophic. It's expanding as you're moving in, as a negative curvature. And we live in a hyperbolic space because trees, for example, outside are hyperbolic. You go from the trunk to the leaves. That's your hyperbolic space. Most of the stem cell differentiation pathways are hyperbolic. You start with stem cells and you arrive at the multiple fates at the end. The beauty is that it's a perfect fit for a lot of hierarchically organized biological data, but also reduced dimensionality. Euclidean space are not very good of handling 1,000 dimensionalities. It's called the curse of dimensionality, and at some point it just can't work. So you can move thousands of Euclidean dimension into about 5 to 10 hyperbolic dimension. Anyway, so to make it story short, we managed now to, with all these tricks, array the epigenetic age or image for these chronologically identical mice between young and old, and they're all distributed and have a number, right? So they're different. But the question you can ask now, how that's related to the function, because for each of these mouse we have function, we have functional experiments. So out of many, I think we had about 15 or 20 different behavioral tests. We arrive at the 9 different clusters because some of the behaviors are collinear, and so you have to kind of bring them together. And so ask the question, how does correlate or inversely correlate with epigenetic function? And it's pretty well inversely correlated. And most of these things are actually positive functions, so it's locomotion. And then if you just correlate it with locomotion, and that's the 3 first clusters that drive the age toward the younger states, then you still have a very good decent correlation. And the locomotion is something that's very fundamental for the life on this planet. If the thing is not moving, it's probably not alive, at least if it's a living animal. Plants are not moving that much. And so that was fairly good. Now, one element here drove it to the opposite direction. We didn't know what it is, but there's a respiratory exchange ratio, very interesting phenomenon, measurements, metabolic. And we found this paper from Rafa de Cabo, of course, a few years earlier, that suggesting that in a cross-sectional study, only old mice and only during the night have increased RER ratio. So that fits squarely into what here we observed. So the conclusion that enlists from those experiments in skeletal muscle, image faithfully estimate biological or functional age. So instead of measuring 20 behaviors, just take this one measurement and this mouse will be old and this mouse will be young. So that fits the bill. That's what you want, right? So you can have a one measurement and you know the function, the locomotor function at least of this animal, or a combination of this function, which we suggest could be a biological age of that mouse with respect to skeletal muscle. Bulk measurement needs to be measured in a single cell. Now, everything I said to you before, it was the cells that we grind the tissue and they basically powder it and dissolve and become nuclei. But that's probably all good in a tissue with this low connectivity like blood, where everything is floating. But it's probably not true for the tissue with a much better connectivity, like this inner hair here again from Roman Hall, showing that this inner hair cells and outer hair cells beautifully connected to the spinal ganglion neurons, and the stria vascularis is somewhere here. So all these elements are connected. If this connection is dead, then both of the cells is dead, and then the connection with neurons is lost. So together with Uri Manor, we decided now to look at this novel, we call it aging model, and a cochlea. And a spatial aging cochlea, I think, is very interesting because it has this amazing property. So inner hair cells distributed along the tonotopic axis. And the position along the axis tells the frequency of the sound that the hair can hear. I'm sure that's not the first time you've heard Jonathan Sebastian, but it's probably the first time you saw how that actually looks like in your inner ear. So the idea here is that you can connect very neatly the function, which is a sound frequency, that cells here. And then epigenetic landscape of this function, because you can dissect cochlea and obtain an image in the whole mounts, each individual inner hair cells with a great precision, and then did all the same manipulation and computation that I just showed you. So, well, there's a couple of vignettes we extract, and this is also the new technique that we develop. I'll show you in a minute. Basically, this is the extracted volume of the cells from this cochlea. That's essentially how every mark, epigenetic mark, including DAPI, looks like. And this is the colocalization, the code tensors, essentially based on the image of the cell, or nuclei, I should say. This is not very clearly visible here. There should be 3 axes, 27 acetylation, methylation, and DAPI. And everything is bound within those 3 axes, but each voxel here computed differently. It's the probability of colocalization, the topology of the system, peripherality close to the nuclear lamina, and a few statistics out of proximity and habitat. And so we found much more effective than autoencoders and deep learning, because a lot of deep learning system models, MLP including, need to be initiated with randomly, and that means a lot of iterations until you find the minimum, where if you know exactly what you're looking at, you don't need that many iterations. You actually directly go to the computation. So this is inner hair cells. We can identify them with a 0.97 accuracy, which is basically certainty. But what we found is that not all features are important. You can actually, out of all the features, which is, I don't know, several thousand features, we can actually attain about 5 to 10%, and those are sufficient. So you start dropping the features, and only when you arrive at this point, you start drop the accuracy. So you can drop 90% of features and still have about 95% accuracy. And you can perfectly isolate the inner hair cells in this UMAP out of everything else. And so what we discovered is the existence of this wonderful gradient. There's a gradient of epigenetic change along basically an entire tonotopic axis. And that gradient can perfectly predict, in a way, position of the cell along this axis. So it fits this nonlinear distribution. And it degrades with age, and we think this is an important feature. You need to have the gradient for effective hearing function. We can separate the cells at a very high accuracy in a single cell level. So this is, each dot here is an individual cell. This is a violin plot. It's a couple thousand cells per plot. These are mice. And you can appreciate there's a 0.94 accuracy separation at the single cell level. So out of 100 cells, 94 times will be correctly attributing cell age. But what's really curious is that when you start to degrade the features and find what the salient feature, the most salient feature of this system, that they start dropping immediately. Essentially, everything matters. There are features that are really important, but every part of the chromatin is affected by aging, as opposed to cell identity, which is really defined by a subset of features, a subset of chromatin state. Everything actually contributes to this aging, and I think this is pretty cool. And we can split them into UMAP clusters that identify this trajectory of aging, and each dot here is a single cell, and there's some clusters intermediately which are much more enriched in a number of clusters. This is HDB scan, so this is a UMAP labeling, coloring by label, and there's a number of clusters in each of this label. So something very interesting happening intermediate, where probably cell destined to die. Now, we trained the model in black 6 mice, right? The model never seen the SUMP8 mice, which is senescent accelerated mice, and this mice things. Basically, they age faster. And indeed, when we look at 3 months, the inner hair cells from these mice are already as old as 9 months. So they accelerated. The aging is accelerated. But interestingly enough, when we treated them with the neuroplastogens, conventionally known as mushroom or psilocybin, this cell gets rejuvenated. You get increase of ribbon synapses and the function, the ABR function, which is interesting, and it dovetails with recently there's a paper in Nature Aging and Belief suggesting that psilocybin extend lifespan of a normal mouse. So shrooms are good, right, for many different indications. Now, the most amazing thing in the system that you can actually measure the function. So the ABR directly measuring the function. At each frequency, the function decreases with age, and so we can start connecting the function to different readouts, what actually is important, which metrics is important for this function. So we look at the cell loss. A lot of time people talk about hair cell loss with age. And it's true, but it's not the first thing, not probably even the second thing, and not the most important things in this experiment that are happening. Although we do have loss in particular 32 hertz, but that doesn't give you universal correlation. If you look at epigenetic age, that's much better. You have not great, but a decent correlation. Now, if you look at tone-topic axis, the score predicted from epigenetic gradient, that's even better. But what's really good is a linear combination of this. And that's much better if you just directly regress it's an IBR. And so this difference will tell you that suggests that different epigenetic differences or hair cell loss in each mouse is different. So each mouse ages in its own different way, similar to DNA methylation linear combination. The combination of this, linear combination of these measurements, provide you the best function. Again, extracting what we hope here, the individuality of the mammalian cochlear aging. Now, in addition to look at little dots, and these cells you can look individually just inside the cell because we use imaging, and the question, what the changes look like in the nuclei. So this is a simple equivalent of the wide field fluorescence. If you just look through everything, it's not very informative because a lot of pixel in front of you. However, if you apply this as a surface rendering, which is similar to 3D contour display, you extract the highest density at each turn. It's like confocal imaging, but you constantly retaking and rotating as you rotate the cell. So what we really been excited and surprised is to observe that there's a huge difference between this 27 acetyl distribution with 1 month and 10 months old mice. It's nice and compact, 1 month, and with age, you're getting a lot of this ectopic pockets, I should say, at 27 high density. And if you overlay 27 trimethylation with the faculty of heterochromatin and 27 acetyl, you appreciate that in young mouse, this marks are very intimately intertwined, but it's completely disconnected in old nuclei. And so we can quantitate this. Indeed, the Betty numbers tell you there's a lot of these pockets of ectopic, which we think activation of the chromatin in an old mouse. And the correlation is really good in young, but completely lost in old mouse between 27 acetyl and 27 trimethyl. And so for the past decade or so, there's a lot of great work been done on different chromatin territories and chromatin domains. But all of this data dealing with the local state of chromatin, the domain juxtaposition. A few years ago, Tom Mestelli, the great Tom Mestelli, provided that review and a cartoon in the review. That's a cartoon. And his thoughts of his imagination, how the different program in cell type A and cell type B would look like. And I think we have the experimental evidence now with respect to aging as a cell state as opposed to cell type, which things probably even more different in cell when you look at the different cell types. So I very quickly go over heterogeneity because it's good with this whole separate subject. But just to tell, if you look at the coefficient of variance and you look at this in the original experiment that I showed you, K4 monomethylation and PBMC, heterogeneity or coefficient of variance increasing with age. And if you do 1 over CV, then you do a straight line and intersect, providing you the maximum lifespan. And it fits somewhere around 44 months for the black 6 mouse. Now, I'm not holding my hands to fire for exact numbers, but it's interesting that 36 is what observed in the Jackson lab, longest lifespan in Jackson lab for 57 black 6 mouse. And it's a wild mouse, any Black Forest, Schwarzwald mice in captivity. So it's a reasonable estimate. And I think it's interesting that estimate coming from this, idiosyncratic dots and speckles. So it's, if you measure it from the, blood counts or steps, it's one thing. But coming from like abstract imaging, I think it's quite impressive. We can do it in different tissues and organs, and it's rather preliminary. A lot of this heterogeneity increases, but it increases not linearly, and depending on organ tissue, things are kind of will be very different. And again, for aficionado, all we do right now is really looking at this complexity, where true heterogeneity is complexity and disparity. And so we're just in this field, and the problem with this is disparity or difference between different cell types. Need the coefficients, so you need to figure out the numbers, how you estimate all these cells are similar, but black and white are completely different, and it measures through the different Rao's quadratic entropy. That's to say that it's a complicated field. But to simplify it, we have this model, the unified model, just to make things fun, that you can position individuals, organs, and tissue and cell on that scale, or the distance, or the epigenetic distance. And within this space, over time, the system will basically progress and eventually collapse on each other. I didn't show this data, happy to talk about it. But with time, the entropy increases, and the faster it increases, the worse is a scenario, because the increase of entropy or variability is directly inversely proportional with the function. The more you vary, the less your functional state is. At some point, the functions gets to zero, and that's end of the game. And that's an interesting association with the cars. If you have the car that doesn't have a proper assembly and it's kind of wobbly, you won't go too far. If the car is tightly built, you'll get an extra miles out of it. So that's, I think, a very good way to look at variance and the progression along the epigenetic landscape. So to very quickly summarize this, I'll just put it out. I'm doing on time. I have just a few minutes. So hopefully I convince you that imaging is quite useful for a number of applications, both identifying cell types and extracting the function, including the function in vivo, and looking at the global chromatin organization. I didn't show any data, but we can do this in vivo. I'm happy to talk about this. But back to the question of what can be done for the elderly. So we're building that initiative where we would like to think about a multi-center study. All of these components are actually people that I've talked to, and we have a lot of strong support from the WA cohorts, of course, where we basically will try to put together really cutting-edge technology to extract cellular, molecular, functional data, including the wearable devices, all the landscape, the imaging that I just talked about, function, and really integrating health condition, diet, lifestyle, social life, and environment. And so hopefully we'll arriving at pretty unique, a comprehensive study, including the biobank, the data sets, and all the technology I've been talking about with hyperbolic space, information distance, and we're building an advisory board. And the more I talk to people, the more people get excited about it. We have a great feedback on how to really initiate and conduct these studies. Have a really talented, terrific group of individuals. A lot of this work been done with former people in the lab. Martin and Han initiated this study. Kanta is our computational guru, a current team member at Perkins, a number of really talented people at UC, at Scintillon in San Diego, and my terrific collaborators. Nothing would be possible without. We acknowledge a lot of mice that contributed to this work, and the funding agency. We also have positions available, PhD students and postdoctoral. Thank you very much. It's a New England fall.
Thanks so much, Alexei. Sometimes a bit difficult to digest, I guess, all the things that are happening. It is obviously an amazing to have you here. Congrats, you're all probably familiar with the fiery distinguished fellowship scheme, which is very competitive, but it's a great opportunity, of course, to bring excellent people to Perth. And we had some candidates we would like to put forward along those lines as well. But fantastic to have you here. Amazing body of work. I think if I can open it up for questions. Oh, the microphone, of course.
I'm not going to sing, I promise. I'll just ask questions. So we know about epigenetics and the importance for quite a while. And I think one point you made, which I always found difficult to put into context is the epigenome response to environmental factors, to what we do, exercise or not, good diet or not, smoking, drinking, etc. And of course, all of these factors we know play a role in longevity and how well people do, how well they age. There's the concept of slow aging. You're doing the right things and you prevent that from happening. And that, of course, reflects in function. Can I ask, so you've done a lot of, let's say basic work in a sense to try to see how you can measure things. Have you done experiments where, for example, you exercise mice or you put them on different diets, which is a lot of interest here in the building, where people are interested in lipids, in metabolism, diabetes. We've got fascinating drugs, and I'm not aware, but it's not my field of expertise. How do these interventions, let's say GLP-1 agonists, which result in dramatic weight loss, for example, are these drugs that are essentially anti-aging? And could you show that with your technique, perhaps even in cells from affected people? And I think that would be an enormous opportunity for collaboration between the campus here, which is broadly focused to a large extent on human and clinical data, but having you, of course, there in the center would raise our game dramatically. Is that something you've done or consider doing?
Right. Yeah, very interesting question. And indeed, a short answer is that pretty much anything you do would affect epigenetics. So the phase of the moon would probably affect epigenetics. In fact, will affect because of circadian. So Neil Einan and Luigi Fontana here, two people that I talked to, and we're collaborating a little bit. I'm trying to extract data from Luigi, which is not always easy. Suggest that indeed exercise and all the diets have a major perturbation of epigenetic. Every time you see gene expression change, you must be assured there's a change in epigenetic, because those promoters and enhancers has to be open and closed. You cannot have a gene expression without a change of the epigenome at that particular locus of chromatin. I showed the doxorubicin and caloric restriction, which is a type of dietary intervention, and there's a massive change, of course. So we very likely would like very much to work with more human samples. So we have some blood samples we would like to analyze. We work right now with the kids. Hospital try to look at PBMC from different immunological interventions. But yeah, that would be lovely. It's exactly what one of my objectives here moving is to actually work with humans, because mice are great, but they don't pay taxes, right? So yeah, I mean, anything, pretty much anything you do, and we know that some small molecules, HDAC inhibitors, for example, you probably can see the effect within hours. In fact, with the topazide, our first data point with, I think, was 6 hours, and it's already maximum response, or 12 hours. So I'm sure within hours we can see a lot of change. I mean, doxorubicin or chemotherapy, terrible drugs, like really killing people and cells and people. I don't know, curing from one thing, but creating such a host of problem that, so that's very effective, of course, and you can see a lot of changes.
I mean, I asked the question partly because it was a very recent publication in the New England where they looked in dialysis patients, and these are patients whose kidneys have failed, who require 3 times a week dialysis, and there's really nothing that helps them, no lipid lowering or anything. But now a study showed that fish oil every day reduced the rates of cardiovascular events by a staggering 45%. So again, this would be an amazing study to do, opposed to placebo, whether or not that, what is the mechanism? I can't explain it. I think the paper couldn't, but so I think enormous opportunities to go down that path. Any questions from the audience, or do we have anything online, Darren? Is there any questions online? Not at the moment. Then Trevor Maury. Trevor has done a lot of work in the metabolic space, also, very much with with assays of the metabolome and other aspects. So another experts here.
Thanks, Marcus. Actually, Marcus stole my thunder with his question, because my question is going to be, how fluid is the measurement of epigenetics? Because
recording in progress.
I didn't say that. So, how fluid is, how variable is it? Because, I mean, you mentioned DNA methylation, which we know, and we've measured it in the RAIN study, and I noticed you had the RAIN study up there as a potential collaborator. We measured DNA methylation when the offspring, I'll call them, generation 2, was 17 years of age, and showed very strong correlations with a number of lifestyle factors. For example, smoking is one of the strongest, but also measures of adiposity and other measures. And then they've looked at epigenetics at 17 in relation to later age metabolic factors and shown relationships as well. But I guess what we don't know is how fluid that is. So is your measurement more robust or less fluid, if you like, than DNA methylation?
I think, so if you talk about fluidity of the epigenetic marks, the demethylation one of the most, I think, more stable mark, and it's hard to change, but also means that it reflects changes at a potentially large magnitude and a longer time scale. It was not that sensitive, although they're doing it in PACE, where they propose to use it as an odometer rather than like speedometer rather than odometer. So some changes probably could be extracted too. So it's just a different way, I think, of looking. So DNA methylation is usually done on large number of cells, and you cannot differentiate which cells you pick unless you do the FACS sorting or any crazy techniques, which just people don't do because it's too expensive and perturb your samples potentially. So, and this is only one epigenetic mark. So I would argue that the imaging will be more sensitive and particularly allow you to go to the single cell level. So you can differentiate what happened in B cells and T cells versus taking the entire blood or entire organ, whatever you look at, and say we change this globally. So you can imagine that your T cells can accelerate aging, but not necessarily monocytes or B cells, or to a different degree. And within the T cells, there is naive and activated, and there's the whole kind of diversity of cell types and responses. And so that all goes unnoticed in the methylation clock. I give you an aggregated value, and it's one number, like literally, and it's a number at distance to the average. Dissect the DNA clock, gives you how far you're from the average. Where that techniques allow give you a distance matrix. So it's a distance everybody to everybody, or every cell to every other cell. So it's a much richer information field that you can extract, including you can look at a distance between the samples. So if you have two people that or two individuals, their tissues and organs, cells like BOT cells, they're very, very similar in epigenetic makeup. Not identical, but within on a scale of few thousand, a few 100,000, those will be like. There's a reasonable assumption then the response of those people to perturbation will be also similar, because if the cells are identical, like twins, you can guarantee the response will be identical. So the proximity between that signatures will enable you to tell something about how they will respond to the intervention, and that's I think one of the key things that is very different from DNA methylation. DNA methylation clocks. Not the global DNA methylation. If you have the whole genome DNA methylation, this of course will be different. But with the DNA methylation clocks, they only measure distance to the average.
Perhaps last practical question. In terms of tissues, obviously we have access, easily to blood samples, but then also tissue from surgeries and biopsies, etc. What's kind of your favorite tissue? I assume blood is very simple to.
I'm equal opportunity employer.
Okay.
I'll take any tissue. Those are very precious, are very valuable. All the tissues, and we can use this as the, frozen samples and grind the nuclei, but you also look. Freeze them appropriately and preserve appropriately, and look at in situ with the sectioning to see the structure of organ and tissue. As I showed you with the cochlea, there's a lot of information, of course, the spatial information that you lose when you grind the tissue and just look at the single cells. But that was very interesting opportunity actually to look at.
Is urine of any use at all? Or there's usually not many
Depends
cells
how many
in
cells,
the urine. That's the problem, of course. Usually not.
Yeah, we're particularly interested in anything that have cells, like nuclei. But then we can associate it and link it and map it onto anything, including plasma, urine, or any liquid biopsies.
Look, if there's no other questions, please join me in thanking Alex for an amazing, talk and body of work. Exciting to have you here, and again, we should really seek active opportunities for collaboration. Alex is probably around for a few minutes as well if anybody's got specific questions. Thanks again also to the entire team of the MRF for the beautiful courtyard. We can't wait for that to be opened. It looks beautiful, and again, I also would like to mention Troy and Zoran, who are really all over the place for us who do clinical research. They really make sure that it does not interrupt our work too much, which we very much appreciate. So they've done a great job. So be kind to them. Give them a bit of chocolate. They love that. So that's Ravi. Thank you so much. Enjoy the rest of the day, and thanks again.